So we're going to continue our phlebotomy videos. In the previous videos we talked about safety and we talked about preparation, and in this video we're going to talk about the quality of blood samples. This is some of the equipment that we're going to be using for taking bloods in this trust. We use a Starsted Monovet system here, which is the same at Addenbrooke's and Ipswich, but if you go to places like Norwich, they might use a vacuum system. We're going to mainly cover needle and holder in this session, so I'm just going to go through the different parts of this now. It comes sealed in a little packet. We have 1.5 inch long needles, which are green needles, so that's the width, denotes the width of the needle. They are covered by this cover initially, but then obviously the sharp bit is inside there. They have a yellow safety device on the side. And then at the back, they've obviously got a part where you connect the bottle to, and inside there is another shorter needle covered by green sort of rubbery material, which will then form a seal when we put the bottle onto it. The bottles themselves are different coloured bottles. We can see all the different coloured here. They have different coloured lids. And if you look at this one, for example, the red one, the lid inside that has another sort of rubbery membrane over here, which would then connect onto the needle, bit of rubbery membrane. And they have these little 3 bumps around the outside. These 3 bumps, they line up to the connecting part of the needle, they push together and they do a slight twist, and then the bottle is attached to the needle, so it won't come off unless you twist it and take it off. If we actually look at the bottle itself, we'll note that the cap, you can actually screw the cap off. Occasionally you'll do that, but very rarely. In your first times, please be supervised for doing that. We'll also notice that when you want to take the blood out of the patient, you have an aspirator, so this actually will then pull the blood out of the vein. You would always aspirate blood for your first bottle, and if you're taking blood out of a cannula. But if you're doing a normal blood test with a needle and holder, every bottle you do after your first sample, you can actually pre-vacuum the bottle, so they'll just automatically put in the correct amount of blood. And in order to set the bottle up to do that, if you just pull the aspirator all the way to the end until it clicks, then it will stay there, as you can see, and you can actually then snap the tail off the end, and your bottle has a vacuum in it, so when you connect it onto the needle, it will just draw the correct amount of blood into it. So that's the equipment that we're going to be using. We do need to make sure that, obviously we've got all these different colour bottles, that we use the right ones, and I'll let Tina talk about the different bottles.
So, Thomas, we've got the different colours, and these are probably the most common ones we use in the trust. We have brown for biochemistry, we have red for haematology, we have green for coagulation, we have blue for blood transfusion samples, and we have yellow for glucose. I didn't use that one. Actually, there is an order of draw with these because they all have different components and chemicals in, and we don't want to contaminate the bottles because that might break the sample down and cause all sorts of problems. So the order of draw is, there is a trick, visualise a tree. So we have brown is the tree trunk, green are the leaves, red is the fruit, blue is the sky, and yellow is the sun. And don't get stressed about it too much. Once you've done it a few times, people just sort of get used to you doing that order, don't they?
It'll be something that you'll get used to. People obsess about, oh my god, I've got the right order of draw, but actually it's something that will just come naturally, and then you can worry about it.
And there is posters in most clinical areas. You can always just ask somebody anyway. But that's a little tip for you.
There are some other blood bottles out there. There's an orange bottle and there's a white bottle. These are very rarely used in the trust, so they do mess up our tree picture, yeah. But they're very rarely used. Okay, so that's the order of draw. Another thing to think about with quality is that we want to treat our blood samples very carefully. So we need to make sure that we try to have the blood in the most natural state as we can. So it's meant to be in the body, but obviously outside the body, we need to be very careful with it. So once when we're using our tourniquet, we don't want to leave the tourniquet on the person's arm for too long. If we do leave it on the arm too long, then the blood can actually hemolyse, where the red blood cells break apart and actually release the chemicals within them, which will mess up your blood sample. Of course, the wrong readings. So tourniquet time is important. You don't leave the tourniquet on for any longer than 60 seconds. Once it's on longer than 60 seconds, your blood can start to become damaged. Other things that can damage blood samples is alcohol. We talked about alcohol in the safety video, that actually if you get alcohol in the cut, it hurts. It has to dry in order for it to work anyway, and if it touches the blood sample, it will ruin it. So you must let the alcohol dry. Other things, you don't want to shake the bottle. I don't know who encourages people to do that, but you don't want to shake the bottle. Just turn it upside down, maybe a couple of times gently to mix it.
Don't put it on a radiator.
Don't put it on a radiator. Just treat it with respect. Once the sample's out, it obviously needs to be labelled, and then it needs to be sent to the lab. And we've got a pod system which we can send the blood samples to the lab with. Okay, so that's everything about quality.
Yeah.
So our next videos we're going to actually be showing you how to set up your tray, and then we're actually going to be demonstrating a blood sample being taken. Thank you very much.
Thank you.